Hi everyone, Dr. Williams here. Today we are going over the axial skeleton. And the axial skeleton, what does that include? Well, it includes vertebrae, the ribs, and the sternum. So we're going to focus on evolutionary trends, the design of the vertebrae and these bones in general, as well as the vertebrate ribs. We're going to talk again about the sternum, and then vertebrae in our caudal fins in general, so if vertebrae in our fish especially, and of course we're going to get into the appendicular skeleton. What we're going to be focusing on here is really thinking about how we structurally need to change to meet the needs for locomotion, okay, in the different environments. So if you're in an aquatic environment, so over here on this figure, you have pressure coming in at you at all sides. And if you've ever like just floated underwater or just gone underwater and then just tried to lay there, you would twist and turn, right? So you would have to do some things to stabilize yourself in the water, but you have pressure coming from all sides. And in the water, you really have to think about drag, okay? So your body design has to deal with drag in the water. So one of the best ways to reduce drag is to have this streamlined body shape. You know, when I used to coach swimming, I always talked about really like, you know, do you want to go this way or do you really want to get into that really tight streamline? So, you know, think about how tight and streamlined you can be. And then again, for orientation, you have to be able to stabilize yourself. So that's what these fins would do, is to help stabilize. But there's more than that too. So it's stabilizing from rolling, from rocking, and then up and down type motions. But when we move on to land, now we have issues with weight bearing. And here we have, you know, basically with force is equal and opposite reaction, right? So as we're standing, our weight is going, the force, our weight, our gravity is, you know, going down into the ground, and we have the equal and opposite forces coming up through our limbs. So now we have to support ourselves for weight bearing as well as increased torsion. So we can't, especially with this weight bearing, if there's torsion is twisting. Torsion is a very, very hard force to deal with because things don't withstand twisting very well. All right, I'm going to make this, I hope, a little bit bigger so that you can see. So multiple times in this lecture, I'm going to want you to actually move. And then when you're looking at the bones, again, look at how these bones will move. So we're going to talk about, you know, in the water, we had fish swimming in lateral flexion, right? So lateral flexion, this is motion in the frontal plane, okay? So this is important to really kind of get this. So when you do lateral flexion, you're moving in the frontal plane, okay? Remember, frontal cuts us in half front to back. Now, granted, I'm using human anatomy terms here, but I think this is the easier way to think about it. So we're moving in this frontal plane. The fish are swimming with lateral flexion, right? Now, one thing that we do see is a switch to dorsal-ventral flexion. So dorsal-ventral, so this is this way. Okay, and we're going to watch this. I have videos coming up that I'm sure I'm not going to be able to keep in here. So you are going to have to click on them because a lot of them are, again, copyrighted. So you can watch them through YouTube, but I will not be able to play them here. So please make sure that you watch and think about these motions. So we have dorsal ventral flexion. If you think about a horse running or the cheetah running that I'm going to show you in a little bit, okay? We actually move in dorsal ventral flexion as well. And you're like, what? We don't do this, right? But we do. So when you think about it, this is also dorsal ventral flexion. So we're moving in the sagittal plane. So if you think about our walking, you know, our limbs are flexion and extension, dorsal ventral flexion, okay? So we're moving into the sagittal plane. So one of the biggest trends or changes from aquatic environments to terrestrial environments is that we start to see this change from locomotion being in the frontal plane, right, to locomotion being in the sagittal plane. Whether you're doing this dorsal ventral flexion, that is going to involve your vertebrae, or sagittal plane with your limbs, okay? If you think about your dog running, okay, that dog, or us when we're jogging, we're moving in the sagittal plane, all right? Okay. Now, here, again, this is exactly what I was talking about. So here we can see, again, this lateral flexion and aquatic. We do see lateral flexion some in terrestrial systems too, so you have to pay attention to that. But in general, you can see these organisms all moving in with lateral flexion. So we also then go from lateral flexion by itself to lateral flexion with trunk suspended between the limbs. We're going to see that in our salamanders on a video in the next slide. And even like a young baby, when they first start to crawl, sometimes you go this way, right? So this is lateral flexion, but you're suspending your trunk. Okay, so you're still moving in this lateral flexion, but now you have your trunk suspended. So we'll see multiple organisms do that. And again, this dorsal ventral flexion is what we see in mammals and archosaurs. So when salamanders are in their larval form and they're in the water, again, they're using this lateral flexion to swim. But when they get onto land, they're suspending themselves, okay, with their limbs, but they're still using this lateral flexion to walk. So they're picking up one limb, putting it forward, then lateral flexion to get the other limb forward. And so you get this back and forth motion, this undulation or movement in the frontal plane. Please go ahead and watch these. It's really kind of cool. All of these videos here, this one, if you move about 40 seconds in, you can save your time. You don't have to watch the whole video. But please go ahead and check these because I could ask you about them. All right, so we just talked about lateral flexion in aquatic and terrestrial. We have lateral flexion with trunk suspended, like we just saw in the salamanders. Now we can talk about dorsal ventral flexion in mammals and archosaurs. And okay, I already showed you again, when you're running or jogging, you're using this, you're running and operating in the sagittal plane. Same thing with this cheetah. Okay, so make sure you watch these. I think this first video is no longer available, unfortunately. That was a really, really good one. But these other two links should still work and are worth listening to. And actually do listen to what the people are talking about. One of it's kind of a little bit fluffy, but it is also talking about, in one of the videos, it talks about the head shape too, and how the cheetah holds its head still so it can focus in on prey. But here you can see this rounded back right here, right? So this is going to be flexion, and then when the cheetah throws out its legs to increase the stride length, that is extension. So it's using this dorsal ventral flexion or flexion and extension to increase stride rate, which helps it run so fast. All right, so hopefully you paused and watched those videos. And now we're going to get into the vertebrae a little bit because we have to think about what's the structure that affects these functions, okay? So some terms. We have a spinospondyle vertebrae. When we think about it, this vertebrae up here is an example of a spinospondyle, and it's a specific type called rhacotomous, which means cut up spine. So basically, if you think about your vertebrae, you could have a centrum, which is this area here. The centrum can be made up of a plurocentrum and intercentrum, and we see differences among taxa which remains, okay? So a lot of times maybe we can't see which is which, but when we have a cut up spine, we can see that the pluro and intercentrum are separated. Then we, of course, have the spines, okay? We also have a holospondyle vertebrae, and this specific vertebrae is a lepospondyle vertebrae. Again, don't get too caught up on these terms. Basically understand the difference though. A spinospondyle, you kind of have this plurocentrum intercentrum, or you'll often see instead of interceptum, you'll see hypocentrum. Or a hollow spine dial would be all of the components of the spine are fused. And this is what we see in tetrapods. Okay, so early tetrapods have this cut up spine, or they're not all fused, but later tetrapods have this fused centrum. And then there's different definitions based on the shape. So the lepospondylus means that these centrum are husk-shaped. And again, you have to be thinking about the notochord. The notochord would have been through here, okay? So surrounding the notochord. Let's take a look at parts of the vertebrae then. So here, we're going to talk about the vertebral body, okay? That's that centrum that I was talking about on the other slide. We're ignoring this other part for now. So this ventral body, okay, or the centrum, provides a lot of strength and support, okay, especially like in mammals. This is a human vertebrae, so this vertebral body is going to be really important for supporting our body weight and distributing those forces of our weight. The size of these bodies are associated with weight bearing. So if you guys get into the lab or see a human skeleton, look at the differences in the size between the cervical, neck region, thoracic, chest region, and lumbar region. Our lumbar vertebrae, the bodies are really like big and beefy, bulky, but again, that's for weight bearing. We talked before about hyaline cartilage. So this edge, there's hyaline cartilage at this, especially at this epiphyseal rim, which is out here, the epiphyseal rim. And this hyaline cartilage permits some diffusion of fluid between the intervertebral discs, and there's some capillaries in the vertebral body. So a little bit of blood flow and diffusion for our discs there. All right, so let's think about form and function with the centra, the vertebral body itself, okay? So we're ignoring all the spines and all the processes. So we're going to start up here with axialis vertebrae. Axialis vertebrae have flat ends. They're very important in distributing compressive forces. And again, if you have looked at vertebrae from humans especially, you're going to notice that this is what we have. So we see axialis vertebrae in mammals. Amphicoelus, so now we're over here. Amphicoelus has a concave centrum. So the centrum in here is concave. It's not fully fused around. So this allows motion in all directions, but it does still limit the motion, okay? And this is what we see in most fish, some salamanders like Nectarus, and we also see it in caecilians. Now, I'm going to back up just a little bit and talk about with restricting motion. So something that you can stand up and do, and this is kind of an easier way to do it. So everybody take, I know you can't see all of it here, but we're going to take our thumbs out, and we're going to have our thumbs out and up, and we're going to raise our hands over our head, okay? So when I do that, when my thumbs are up, I can actually do this full range of motion, right? Now, I want you to go ahead and rotate your arms in, okay? I'm going to do it with just my left hand so that maybe you can see it. Rotate your arm in, and thumb is now pointing down, and go ahead and lift your arm and see how far you can make it go. You can't make it very far, okay? That's because bone is hitting bone. There's what's called, if you're a physical therapist, or you might hear the term hard stop. So bone against bone is going to restrict that motion, and that is a hard stop. Now that's important because, we're not very effective when we're lifting like this. If we're going to lift something over our head, we're going to do it this way with our thumbs up. Okay, so same principle with these vertebrae. When you're thinking about how these vertebrae articulate, what kind of motion are they going to allow and what kind of motion are they going to restrict? Okay, I'll make myself smaller again. So now again, we can see this amphicoelus, and you can imagine with that kind of concaveness that, yeah, it would allow dorsal ventral flexion a little bit. It would allow some lateral flexion, probably a lot more lateral flexion, right? So moving in this frontal plane. But it would still limit some motion. There's only so far you can go. All right, another vertebrae that we're going to look at is procoelus, okay? This is where you have concave anteriorly, so this is the anterior side, and convex posteriorly. And then we have the blue is the intervertebral disc. So this allows a ball and socket type motion. So another joint that you have as ball and socket is your shoulder joint. So again, if you think about when you stand up and you move your arm, this ball and socket joint with your humerus to your pectoral girdle allows a large range of motion. Okay, so I can do all kinds of things with that. So get up and move a little bit and check it out. So anyway, so this ball and socket type joint, now of course they're vertebrae, so there's not going to be as much range of motion because there's tissue around it that is going to limit it. But in general, it does allow more movement. And this is what we see, this procoelus is in anurans and extant reptiles. We're going to go to the next slide now to go over the rest of these. So now we have Postcranial, which is right here. And this is just the opposite. So it's still a ball and socket. We can see this ball and socket type joint. But we have concave posterior and convex anteriorly. Okay, and this is what we have in most salamanders. Then we also have heterocoelus, and this is a saddle shape. So we have lateral and vertical flexion, so lateral and then vertical, this would be the dorsal ventral, but it prevents torsion or rotation, okay? So it prevents this twisting motion, okay? So we see these kind of vertebrae in turtles. That helps turtles retract the neck down into their turtle shell, okay, so that they can rotate it down in, but it prevents it from twisting. And same thing with birds and their cervical vertebrae. So birds have a lot of motion in their head, but we don't want that torsion. So these vertebrae help to prevent torsion. All right. Now we can get into a little bit more information with our central functions. So when we think about axis of rotation, axis of rotation is perpendicular to the plane of movement. So again, I'm going to see if I can do this. All right, so if I'm going to do it with this hand, this is flexion and extension. So when I do flexion and extension, my axis of rotation comes through here. So if you're thinking about a hinge, right? If my elbow is the hinge of a door and my forearm is the door that's moving. So the axis of rotation is going right through the middle of that joint. So when we look at vertebrae, with acoelus and amphicoelus vertebrae, with their centra, the axis of rotation is actually on the rims, okay? Now this is a problem because it tends to stretch the centrally located dorsal nerve cord, remember? So the dorsal nerve cord is up here behind it. So it's not a huge deal because acoelus and amphicoelus aren't using a whole lot of motion in the sagittal plane. But if they did do a lot of motion in the sagittal plane, then they could be stretching their nerve cord, which could be damaging. But when we look at procoelus and Postcranial centra, again, these ball and socket ends, they have the centrally located axis of rotation, and that limits the damaging stretching that we see. All right, now the rest of the vertebrae. So we talked about the centrum, or the vertebral bodies. Now we also have some arches. So we have a neural arch and a hemal arch. Now you're not going to always see these hemal arches, okay, because they're not being used anymore, so we don't have them in a lot of taxa. But in fish, we definitely see this. So we have the centrum, we have a neural canal. Through here is where the spinal cord is running, the neural spine around it, and again, the hemal canal and the hemal arch. With the centrum, again, remember that the centrum, this is surrounding the notochord. All right, so here we can look at a human vertebrae. again, we have our centrum or vertebral body. We have the neural arch back here. So this is the neural arch. With the neural arch, we have the pedicel, the lamina, and then we have all these processes, okay? So again, this pedicel and lamina all surround this vertebral foramen, which creates this vertebral canal. And again, what structure passes through this canal? So that's an important one to remember. Now, one of the things, if you've ever heard of a laminectomy, that's what they do to release pressure on the spinal cord, basically. That's what's running through the vertebral canal. So this is where you would cut the lamina here and then remove or make more room. And we used to do that on human cadavers. I'm not sure if there's a current human cadaver with a laminectomy so that you can see. I've done it on a cat before, and it's really, really tricky because it's so small. So maybe when we get into the spinal cord and nervous system later in the semester, you'll get to see that. All right. So now with the vertebrae, okay, there's all these bumps and processes, and we do have to pay attention to these because these bumps and processes, or apophyses, okay, these are processes that project from the centra and the arches. So again, the centra here, and again, we have the neural arch posteriorly. So when we think about diapophyses and paraapophyses, these are what are articulating with the ribs. So here on this human vertebrae, right here and right here is where the rib would articulate. So the dorsal rib would come here and then touch here and then come around and help protect ventrally. The base apophyses are they're paired ventrolateral processes, and they're remnants of the hemo arch. So down in here, if you have them. Okay, so a lot of times we don't see those depending on the organism we're looking at. So here, this is a necturus vertebrae. And here again, we can see the centrum, vertebral body. We can see this neural arch. We have zygapophyses that we'll talk about later, and here are the diapophyses. So these are the articulation points with the ribs. All right, so now we can talk about these. Again, these apophyses or outgrowths, they do help to limit movement. They're also important for attachment sites of muscles and ligaments. Again, parapophyses are early on the hypocentra and the demifacets for the dorsal ribs, okay? So, all right. One of the things we're going to really think about, though, is the pre and post zygapophyses, okay? And that's these parts here, okay? Here and here. So here's a pre and here's a post. So we can come over here and look at what I have circled in this center here. And this would be a post if this is anterior part of the organism, and this is the caudal end, the posterior part. So this would be a post-zygapophysis and the pre-zygapophysis here. These articulate, and you can see that they're going to limit motion. So just by looking at this in this articulation, you should be able to see that it's going to prevent this dorsal ventral. Okay, those bones are going to hit each other if you try to flex and extend dorsal ventrally. But it looks like these would probably slide on each other if we did some lateral flexion here. Okay, and specifically these are going to prevent torsion. So if you try to twist the vertebrae, those two bone projections are going to limit that rotation. All right, so here again, here we can actually see some of these basopophyses here. These are the chevrons, remainder of the hemo arches here. Here we can see on this fish again the dorsal ribs. So when we're thinking about the parapophyses over here, our dorsal ribs are coming off of them, and then here with the fish we also have ventral ribs. Okay. So again, in tetrapods, we maintain the dorsal ribs but lose the ventral ribs. Okay, so going into this a little bit more, again, with these apophyses and these articular facets. So articular facets is here, this kind of in blue on this. This is where there's an articulating apart. So again, the shape and the direction, the orientation of how they connect to each other determine the movement. And there's going to be different parts of the vertebrae, again, different regions that have specialization in different shapes that allow function and movement in those. Again, the zygapophyseal joint, there's pre and post, or cranial and caudal zygapophyses. Here again, this is on the human. Here we have the intervertebral foramen. This is important because this is where your spinal nerve is coming out. Okay, so you have your spinal cord running up through the vertebral canal. That's going from head to tail there. But you have to have nerves coming out. And this intervertebral foramen is really important because if you crush that, then your nerves die. Anyways, so here again, we can see this on a necturus. We see the centrum. We see the neural spine. We see the zygapophyses, so the connection here. We also see back here, they've removed the rib here, but here we can see the rib and how the rib is articulating with this. Okay, so now we're going to talk about the differences in the levels using humans because we can actually move. So in our cervical region, this is a cervical vertebrae. And these zygapophyses that I was just talking about, so pre and post. So this is a pre-zygapophysis, okay? So here in human terms, we're calling it superior because it's towards the head, but this would be our pre. And if you think about that, that's really kind of flattened, okay? So in our cervical region, because it is flattened there, we can actually move quite a bit. So go ahead and do that. We can turn our head this way, in the no, we can do yes and no. So we have a lot of motion and flexibility. So yeah, you know, we're sitting too much. Go ahead and move your head around. But that action is made possible because these superior articular facets are kind of more flattened, okay? So they don't restrict as much movement there. This is a thoracic vertebrae, okay? So if you think about our ribs area, our thoracic region, these zygapophyses, here we can see the superior articular facets, or the pre-zygapophysis here. And these are more vertically oriented, okay? So if you think about it, if you have the orientation here and you try to do flexion and extension, it's going to stop it, okay? But it will allow movement around. So as you're sitting here in your chair, go ahead and sit up nice and tall and do a nice spinal twist. Turn around. Think about where that movement in the turning is going, not just in your neck, but in the torso here. If you feel down in your lumbar region, that lumbar region isn't twisting, but your thoracic region, you have little bits of twisting around because that the zygapophyses in the thoracic region allow that twisting. In our lumbar region, we can look at our zygapophyseal joint here. And now these joints, or these bony projections, kind of project out to the side a little bit. So basically that's going to prevent that rotation. So when you went ahead and did that rotation, you notice that, hey, I didn't really rotate in my lumbar region, right? But this does allow a large amount of dorsal ventral flexion. So when we do this dorsal ventral flexion, that is mainly occurring in our lumbar region, okay? And then here I have a sacrum, and again, the sacrum, you can see this fusion is going to really limit the motion, okay? And this is going to be very important when we think about weight bearing. Okay, so I just wanted to kind of go back and think about our joints of the vertebral bodies first here. So here we have our centrums, okay, or vertebral bodies. We have the nucleus pulposus. So remember that this nucleus pulposus is a remnant of the notochord. Hopefully you've got that in your head by now. So basically these discs are really important for shock absorption, okay? These joints are called symphyses, and they're secondarily cartilaginous joints. We have these intervertebral discs. Again, mammals have this notochord here, and we have cartilage in between, okay? So this again is going to allow different, compressions. So especially in mammals, when we have this flat surfaces on both sides, so it's going to allow distribution of weight across that whole surface, and it allows anterior and posterior flexion as well as lateral flexion. Looking into these intervertebral discs just a little bit more. So this is the disc right here. We're going to take it out and look at it just a bit. We have two parts of the disc. The outer part is the annulus fibrosus, and this is really strong fiber cartilage. And they're made in concentric rings, and the way the cartilage is laid down is in perpendicular to each other. So one layer comes this way, the other layer is perpendicular to that, and that helps really increase the strength of that. These, again, insert onto the epiphyseal rims of the vertebral body. So if you look at a vertebral body, or the center, the outside of that, those epiphyseal rims is where it's actually really attaching hard to. These do thicken with age because, again, fiber cartilage, it will dry out with age, so it tends to get more cracks in it. Here, okay. Oh, another thing I really want to note is notice how our nucleus pulposus, the center is posteriorly. It's not centered in these vertebrae, okay? So yeah, usually if we get a herniated disc, because this is more posterior, usually that herniated disc goes posteriorly. All right, so ligaments. So ligaments are to restrict motion as well. So we have hard stops with the bone-on-bone stops. Ligaments are some of the things that help with soft stops, okay? The tissue that helps to contain movement. So we have all these ligaments that help to limit movement between the vertebrae. They're also elastic in nature, so they help us conserve energy. So we don't always have to have muscles to hold us there, but they kind of help us store and conserve energy. We have long ligaments that go the whole length. So here we have this is the ventral part of the centra here. So we have this, again, in human terms, anterior or ventral longitudinal ligament. This is running the whole length of your vertebral bodies. So if you just looked at this and didn't dissect it out, you would have this coming up the whole length of your spine. You also have a posterior one. So here you have this posterior longitudinal ligament going the whole length of the spine. So these are going to limit motion. So when I do flexion, this posterior longitudinal ligament is going to limit how far I can flex forward, okay? And it's going to, as I stretch that like a rubber band, it's going to have some elastic energy to help me come back up, okay? Same thing with anterior longitudinal ligament. So if we're doing extension of our back, if we extend back, that anterior longitudinal ligament is going to be stretched, and it will limit how far we can go back. We have a lot of little short ligaments between everything too, so from the processes all over the place. We have all these short little ligaments as well in between. In here, the ligament of flavum, don't get caught up on all these names, but all these short ligaments are also more elastic, and they store energy when stretched, and again, release this energy when it's relaxed. All right, ribs, okay? So embryologically, ribs preform in cartilage within the myocepta. So you think about these segmented body plans during development, and the ribs are growing in this, in the myocepta, okay? With agnanthans, we see no ribs. In fishes, we have dorsal and ventral ribs. In sharks, they have dorsal rib only. Tetrapods, right here, a tetrapod, they also only have dorsal ribs. And we see dorsal ribs in the trunk region, and we tend to lose them in the lumbar region. So with birds, they also have these uncinate processes. So here, these are the dorsal ribs that are retained in birds, and these uncinate processes help to overlap. So again, a hard stop. So it prevents, like when the bird strokes down, it prevents the muscles from crushing in on the ribs, so that you don't crush the thoracic cavity where your lungs and heart are, right? So it helps maintain support there. So Our ribs can be used in locomotion, okay? So we have muscles attaching it. We have muscles up here that attach to our ribs, our pecs, right? We might do bench presses or push-ups or all kind of stuff to develop our pecs. We don't use our arms necessarily for locomotion, but if you're thinking about brachiation, or if you're a four-legged or a dog, say, they are actually using these muscles for locomotion and attachment sites of the muscles onto the ribs. They're also involved in respiration. So we have muscles within these ribs that help us respire, okay, help to increase and decrease the thoracic cavity volume. As well as a function I don't have on here is protection. So again, we're protecting our heart and lungs. Okay, with the ribs and sternum in general, the sternum then is this mid-ventral bone, okay? So sternum right here on our crocodile, and here we have a sternal plate in salamanders, so reduced here. But embryologically, these are endochondral bones, okay? Endochondral bones. So they have a cartilaginous template, and then they grow and ossify. This is, again, the origin and site for chest muscles, like our pectoralis is on here, and some other muscles as well. It helps to secure the ventral tips of the true ribs. Okay, so again, the ribs are attaching here, okay, or articulating with the sternum, and this helps to protect the rib cage. And again, with the sternum, it can either be a single bony plate, like we have in salamanders, or it could be several bony elements, like we have 3 elements for our sternum, which I'll show you in a minute. But here again, here's the crocodile sternum. Okay, so fish and early tetrapods lack a sternum, okay? Again, if you're thinking about locomotion on land, that's where you need that sternum for those muscles. But fish and early tetrapods do not have a sternum. Again, Urodels, salamanders, up here, a single sternal plate. With anurans, they get a xyphosternum with a xiphoid cartilage. Okay, what in the world am I talking about? So we can do on us. So I'm going to come up here. This is a young monkey, but we can feel ours because we are, monkeys. Up here at the top, we have the manubrium, and there's a notch here. So this is the manubrium. Then you have the main body of your sternum, and then lower down, you have the xiphoid process. So if you ever took first aid where you had to do cardiac compressions, you're supposed to come up so far. You have to be careful not to go too far down because you could hit the xiphoid process. So in mammals, like we have these 3 bones, the xiphoid process, the main body of the sternum, and the manubrium up here. So when we're thinking about anurans, they have a xyphosternum with a xiphoid cartilage, okay? Not a good picture of it here. In reptiles, the sternum is absent in turtles, snakes, and many limbless lizards. But other reptiles, we see this single mid-ventral element associated with the pectoral girdle. So here we can see this in a lizard. And birds, of course, have a birds that fly have this enlarged sternum or a keel for muscle attachment. And I already went through mammals. So, sorry, I don't have a real copy of this image, so I had to take a picture of it. But I really like this because it shows us these evolutionary trends in these vertebral elements, and that's some of the things that we're going to really focus in on. So we're going to focus in on each of these separately, but I want to kind of go over here. So we can think about, this is our notochord, okay? In our cephalochordates, we have our notochord. And then in a protovertebrate, then we start to see some bony elements surrounding this notochord, right? And like the lamprey, an ancestor would have had some bone, and there's some question marks on this. We can see, again, the ostracoderms, we start to see some bony elements around our notochord. And then here is where we see the evolution of hemal and neural spines, okay? And so here we can look in a placoderm. We have hemal spines and neural spines here. Then we can see again in chondrichthyes. Okay, this is very, early on we might have had these kind of dorsal projections, and later in sharks we have this cartilaginous area. Here then we get these vertebral centra developing here. And here we see with our teleos. So our teleos fish are all going to have centra, as well as all the tetrapods. Rhacotomous vertebrae here as a synapomorphy with the sarcopterygians and tetrapods. So we see this rhacotomous, or this cut-up vertebrae occurring here, and then eventually we see fusion later on. So we're going to see, once we develop these vertebrae, we're going to see loss of these intercalary or interneural arches, okay, like bony elements in between these arches. We're going to see the loss of the hemoarches. So hemoarches, here we have them in fish, but we see a loss of that in tetrapods. We see the origin and variation in the number of centra. So again, whether it's cut up or it's fused. We see fusion of components, of course, and then, of course, regional specializations. All right, so what in the world am I talking about with loss of intercalary arches? So we're going to start over here with a primitive fish. So here we have the notochord. We have the dorsal nerve cord, okay? And we start to see these bony elephants, so neural arches, hemoarches, so dorsal and ventral components. With these dorsal arc components, we have these dorsal intercalary components, okay, in here. Those will eventually be lost. So over here in our salmon, our teleos, we see that we no longer have these dorsal intercalary components. And we also have ventral intercalary components. So again, we have the hemoarches, and in between these hemoarches, we see these ventral intercalary components on primitive fish, okay? When we look at our chondrichthyes, we see again, we have the centra, the vertebral column in here, okay? This would be the bodies, but again, remember this is cartilaginous. We have, so then this is around the notochord. Here we have the neural or vertebral arch. You're going to see vertebral arch a lot. So vertebral and neural arch are synonymous. I do like neural because it does get us back to it's surrounding the spinal cord or the dorsal nerve cord. So here we have the neural arch, and then we have dorsal intercalary plates here. So these are the plates that we have. And again, in the teleos, we lose those components. So here we're going to get into this a little bit more, looking at these. Again, the cartilaginous fish, we kind of just went over that. So again, we do have these neural arches with dorsal intercalary plates between the arches. And actinopterygii, so this is our chondrichthyes, sturgeons here, our paddlefish and modern lungfish have this pattern. So we see this incomplete centra. So again, here's the notochord. We don't have fusion of these elements, so it's incomplete centra around the notochord. The notochord is not completely constricted, okay, because it's not completely surrounded by bone. Cartilage is deposited into the notochord sheath, though, that helps with increased structural support. Then when we get into teleos, so here we have teleos vertebrae, and here we have amia, or the bowfin. Okay, amia is the bowfin, so I put a picture in here because they're really cool looking. But here, we lose these intercalary arches. We have ossified amphicellus vertebrae. Okay, so ossified amphicellus vertebrae, our centra are fused here. The centrum, it becomes this major mechanical support, okay? The notochord, however, does persist within each centrum. So, it's still, the notochord is still there, okay? So here again. All right, so now we're going to talk a little bit more about this fusion of the elements. And this figure, the coloring is a little off, but the cross-hatched areas are hypocentrum, and it says black. It's actually kind of reddish here. These are the plurocentrum. So we're going to talk about these centra and the fusion of these elements. So again, in early tetrapods, we see multiple segments, right? Just like we see in the fish, we saw multiple segments. And our crossopterygians, again, we had rhacotomous vertebrae. And again, we usually see these large wedge-shaped hypocentrum and then smaller pleurocentra. So again, the hypocentra here are larger and we have smaller pleurocentra, okay? Now the trend in amniotes, though, is that we see the pleurocentra. So this part, the pleurocentra, ends up increasing in size and being a larger component in this. So here we have a primitive reptile. This pleurocentra is a larger component, and this hypocentra is reduced in size. And then our modern amniotes have this centra is made up of the pleurocentrum, okay? All right, in the temnospondyl lineage, the interseptum tends to predominate. Interseptum is another name for hypocentrum, okay, because you need more names for things, right? So you have to think about what are they talking about on these different figures. In that Anthracosaur lineage, we saw this pleurocentrum. Now, what am I doing? I'm down here. We can kind of see this. So this is Rhipidiscians here. We have Ichthyostega, and here we have our temnospondyl lineage. We have Anthracosaurs, the lepospondyls, and then amniotes coming off from a common ancestor in here. So in general, we see this trend towards fusion, right, and decreased flexibility to handle increased compressive forces that are associated with being on land. Okay, so that's when the trend, we see fusion of these elements, fusion of the centrum, and that's because we need decreased flexibility to handle increased compressive forces. Okay. Cool. Now we can talk about regional specialization. Okay, so again, we can see these trends. Okay, so here we have a bony fish. In a bony fish, you have two regions. You have a trunk region and a tail region, trunk and caudal. When we get to this labyrinthodont, we can see a little bit differences. We see a kind of shortened cervical region. Again, that heavy bony plates on the back of the head that's going to help to or limit movement, basically. But we have a cervical region. We have a trunk region. That's thoracic and abdominal together, this trunk region. We have a sacral region. The sacral region is going to be really important on land because, again, we need to think about our pelvic girdle and how do we do weight bearing on land. So we see the sacral region here and then a caudal region. In a primitive amniote, again, we're thinking about more time on land. We're going to have more specialization. So now we have a cervical, we have a thoracolumbar, we have a sacral and a caudal region. And then in early mammals, this is where we start to see the differentiation that we actually have. So we have a cervical region, a thoracic region, a lumbar region, sacral region, and caudal or tail region. All right. So when we're thinking about vertebral columns and tetrapods, again, we have cervical, thoracic, lumbar, sacral, and caudal region. In the cervical region, we're going to start there. Amphibians, they have a single cervical vertebrae, and there's not a whole lot of head movement. If you watch a salamander trying to move around, it doesn't move its head a whole lot. So single cervical vertebrae, not a lot of head movement. Reptiles usually have 7 cervical vertebrae, and this allows for increased flexibility. Birds really have a variable number of cervical vertebrae, and up to 25 in swans. That's in contrast to mammals, so we kind of have that reptile pattern. We have 7 cervical vertebrae, whether we're a human, a monkey, a dog, or a giraffe with a really long neck. 7 cervical vertebrae. In reptiles, birds, and mammals, we see the first 2 cervical vertebrae are an atlas and axis. So here I have a human atlas, that's the cervical vertebrae 1. The atlas is going to articulate with the condyles of the skull. So remember we talked about the occipital condyles. So in mammals, we have 2 occipital condyles. Okay, so the atlas has these 2 articulation points that it's going to articulate with the occipital condyles of the skull. And then the second vertebrae, cervical vertebrae, is the axis, and it has this projection called the dens that sticks up, and the atlas rotates around it. Okay, And in birds and mammals, we also have in the cervical region a transverse foramen over here, and this is for the vertebral artery to come through. So the vertebral artery is an important secondary source of blood supply to the skull and brain. All right, we're going to talk a little bit more about the cervical region and specialization in there. So again, cervical vertebrae is really important for moving our head, okay? Cranial mobility, moving our head. Again, we see more in mammals and birds. We see reduced or no free ribs. So in the cervical region, notice that we don't have ribs. So these were cervical ribs in this skeleton, but they're reduced. Here in the skeleton, we don't have any ribs in the cervical region. Again, in humans, we don't have any. Again, we have specialization of the C1 and 2 vertebrae, the atlas and axis. Again, and I already talked about that, and the articulation with the skulls. They don't have any, the presycophyses, there's not sticking up projections, so we do have motion with yes and no. With the skull, we also see transformations here. In fishes, we see unpaired non-articular occipital processes, and again, tetrapods, we either have 1 or 2 occipital condyles. So again, mammals, we have 2, birds have 1, so go ahead and go back to your notes from last week and make sure that you understand what taxa have 1 or 2 occipital condyles. Now we're going to get into the thoracic and lumbar region, okay? So again, in the thoracic and lumbar region, here, thoracic and lumbar, we see in fishes that we have a trunk and caudal region, okay? So the trunk region looks the same throughout, and then we have a tail region. In amniotes, again, we have a thoracic region, and we have dorsal ribs, okay? And we also have a lumbar region, which is our posterior trunk vertebrae without ribs, okay? So later amniotes will not have ribs there. So when we're looking at this, and again, with our trunk region, fish, amphibians, and snakes, the vertebrae between the cervicals and the sacrals, okay, that's all that area, and they articulate with ribs. This is just what we call the dorsal region, right? But in crocodilians, lizards, birds, and mammals, again, we have ribs on the anterior trunk region, so the thoracic region, up in here, thoracic versus lumbar. But we lose these lumbar ribs, okay? So see here we have lumbar ribs here, but we do not have lumbar ribs here. So I want you to pause and think about, why do we lose ribs in the lumbar region? So pause it. When you go to the next slide, we'll talk about it. So it has to do with our locomotion. So if our locomotion is in the sagittal plane, we're using dorsal ventral flexion. You can imagine that if this dog had lumbar ribs, it would get in the way of our dorsal ventral flexion. Okay, so we see this loss of ribs in this lumbar region that helps facilitate our locomotion on land in this sagittal plane with, again, dorsal ventral flexion. Okay, so regional specialization in the sacral region. Again, this has to do with providing a solid support for weight bearing with our lower limbs. So we see fusion in the sacrum or synsacrum. With the sacrum in general, the sacrum is a single bony complex, and it's basically a bunch of fused sacral vertebrae. Okay, so in the human, this is all our sacrum, and then we still have a tail. This is our coccyx, or tail. Here we have this fusion of elements called the synsacrum. We can see in Nectarus, the frog, we see fusion here, a cat and a hamster of all things, as well as an alligator. We still see some fusion of these elements in the sacral region. And the sacral vertebrae, they tend to have these short transverse processes, the sticking out part. They help to brace the pelvic girdle and the hind limbs against the vertebral column. And again, you have to have a solid support. You can't have a lot of bony elements, or else you're not going to have a lot of stability when you try to apply forces. These are fused with the ribs and the attachments to, again, the pelvic girdle. In amphibians, they have one sacral vertebrae. Amniotes usually have at least two fused with the ribs. Our living reptiles and most birds have two sacral elements. So here we have alligators with two sacral vertebrae fused together. And again, most mammals have 3 to 5 that are fused. And you can kind of see that. Here's one, here's another, here's another. So you can see that fusion of those bones. All right. So now, caudal region, the tail region. All vertebrates have a caudal region, and there could be hemal and neural spines on them. Primitive tetrapods had really, really long tails with 50 or more caudal vertebrae. If you look at some of the dinosaurs, they had really, really long tails, caudal vertebrae. In extant or living tetrapods, we see a reduction in the caudal vertebrae in general. We also see that these arches and the processes get shorter. That also means that it tends to taper. The tail tends to taper at the end, which makes sense. The smaller at the end, less weight to have to lift it by. In anurans, they have a really cool modified tail. They have this terminal segment called the urostyle. So right here is their urostyle, and here's our coccyx. All right. With the caudal region, we can talk about it a little bit more in birds and dinosaurs. You start to see, at least in some dinosaurs, like here's the oviraptor, you see this fusion of these caudal vertebrae becoming the pygostyle that we see also in eagles and here. Again, just to remember that we're talking about in this area. And here on this cute little hummingbird, it shows you this. So in apes and humans, we have the coccygeal or tailbone, we call it. And it's that fusion of the last 3 to 5 vertebrae. Now I'm going to make this a little bit bigger because I think it's really cool to think about. So this little hummingbird that we're holding, that we captured, say, at my house, we can actually see that these tail feathers are on this caudal region, and they're all attached to this pygostyle. So there's little muscles that help to erect it and change the shape, that kind of stuff. So that's that region. Now, this slide, you're probably like, oh my God, there's so much stuff on this. If this helps you to put everything together, that's great. If not, don't worry about it a whole lot. But here we can actually work through this. So we have regional specialization. We have a cervical region, okay? Goes from like one cervical vertebrae to multiple cervical vertebrae. The more cervical vertebrae, we have cranial mobility. We have, again, more in mammals and birds. We have reduced or no free ribs. We have transverse foramen in birds and mammals. And we see this rib fusion, okay? So this is where those hemal arches would have fused and allowed this transverse process. Within the cervical region, we have specialization of the first 2 vertebrae, the atlas and the axis. Okay, and some summary there. And again, we can think about how the atlas, the first cervical vertebrae, articulates with the skull, whether it's an unpaired occipital process in fishes to 1 or 2 occipital condyles in tetrapods. We can come up here and think about our trunk region. In our trunk region, we can think about our thoracic region and our lumbar region. Okay, some have no differentiation, like here. We have just a trunk region that's not differentiated. And then in others, we have in amniotes, we have a thoracic and a lumbar region differentiated. We have our, again, sacral region, okay? And in amniotes, we have some of the summaries here. We also have, again, our tail region, our caudal region, and some of the things that go along with this. So this is just kind of putting everything I talked about on the previous several slides into one figure. Now we can get into fins and caudal fins. So here we can talk about different shapes. Okay, so here we have a heterocircle tail. This is also a heterocircle tail. Here we have a homocircle tail and a diphycircle tail here. Okay, let's go through some of these. And why do fish have different shaped tails? So a heterocircle tail is where we have the vertebral column. So here's the vertebrae or vertebral column. And that tail vertebrae end up going up into this dorsal lobe of the tail. Okay, and this again is what we see in sturgeon, polydon, that kind of stuff. So sturgeon, polydon, we see this dorsal or heterocircle tail. We have in the diphthocircle here, we're skipping down to diphthocircle. This is where the vertebral column goes straight down the middle of the tail. Okay, it's symmetrical on both sides. Okay, the dorsal and ventral fins are symmetrical. And this is again where we see it in Latimera. All right, then we see homocircle tails. So this is what we see in Teleos, for example. And here, this is where the vertebral column slants up to support the dorsal edge of the fin. So it's slanting up. It's not continuing up as much into the dorsal fin as it does in heterocircle tails, but it still is slanting upwards. And we keep these hemal arches. So these hemal arches are sending down these, the hemal arches actually act as a supportive struts. So that's what we can see in, say, Elaps, for example. Okay, so why in the world am I talking about this, right? Well, it has to do with function. So the tail is helping with thrust and lift. All right. So if we think about this, if we see this shark swing around and we have this heterocircle tail, the dorsal lobe is deflecting water down, producing lift, okay? The ventral lobe is going to do this upward deflection of water that would make the shark or fish go downward. So here we can show in this figure, I'll kind of walk you through this. So in an intact shark tail, basically you have this dorsal lobe that's deflecting the water down, creating lift, so the force is going up, and it also is producing thrust. We have this smaller ventral lobe that helps us from going up too much. And we can see that if you remove this ventral lobe, okay, and this dorsal lobe is producing all this lift, the force is going to go upward more, okay? So this ventral lobe is really important to help with, leveling out so that we can go forward. So our force is this way, okay? So we get lift and thrust forward. If we remove this ventral lobe, we would just be lifted up. The nose would be lifted up too much. Okay, so we have a sand tiger shark up here. We have an Anapsida here. You can again see this homocircle tail, so it's just producing thrust so that the fish is going forward. We have again a heterocircle tail here, producing thrust so that we're going forward. And here again, more thrust and lift. Okay, the more divided this heterocircle tail is, you're also getting more lift. Now, this ostracoderm, this Anapsida, why in the world is this lower lobe, the ventral lobe, larger? Okay, so think about that. What would that do? And how does it affect the ecology? Well, hopefully, if you remember when we talked about ostracoderms, we predict based on their morphology that they are bottom feeders. So this larger ventral lobe, this hypocircle tail, is going to help them keep their nose pointed down so that they can feed. All right, so on this figure, we're going to kind of go through it rather quickly, but I left all of this text on here for you so that you can kind of go through it more closely later on, just to make sure that you get it. So when we're thinking about the spine, we can think about the forces that are applied for it in the function. So when we think about the fourth bridge, okay, it's going to resist compression, okay, so resist being pushed together too much by the rigid members, okay, and it's going to resist tension by these more flexible members. So each section of the bridge is resting on these piers here, okay. So here in this figure, we're going to move the piers combined apart, okay. So the weight of the railroad here can span a distance between the nearest piers, right? So all of this. And when we think about that, we're going to have these pulling apart or tension being pulled apart, and the forces are pulling this way on it. All right, so why in the world am I talking about bridges? When we think about it with the spine of vertebrates, we can see this basically the same function. So the forces are being pulled out this way and the forces are pulling out this way. So we could take these same arrows right here and put them down here. there's a little star here. So that's the trade-off in weight distribution between these two piers, right? So this in here, this would be like where the weights, if you're just on this side, the force is going to be pulling that way, and opposite here. So this is the nodal, okay? So basically, again, you can actually look at vertebral columns of different organisms, and you can see the difference in these spinous processes. So notice that these spinous processes are kind of pointing caudally, and then when we get down here after this inflection point, we see that the spinous processes are pointing this way. This is going to be important when we start thinking about our muscles and the ligaments attaching to that and how they're pulling to help resist these forces. So big take-home points are in bold here. So bones are going to resist compression. The muscles and ligaments are going to resist those tensile forces, those pulling apart forces. And the change in orientation of these neural spines is going to mark the nodal point where these forces differ. So again, it's a lot of fun. Hopefully you can get in the lab or look at different pictures online of the vertebrae and see if you can see that inflection point. Okay, same thing when we're thinking about how we hold up these arches and what's going to help resist. So we have, ventrally we have an arch that comes this way, and then in the cervical region we have an arch that goes the other direction. So again, we have muscles and ligaments that are going to help hold that arch, okay, just like they're talking about with a violin string or something like that, or a bow. When we get into balance, and again, thinking about bipedal balance, we can think about this cantilever. So again, when we saw a lot of these dinosaurs, okay, or even early mammals, we saw these really long tails. And basically, when you start to get to this upright posture into a bipedal balance where you're using your hind limbs for locomotion, freeing up your forelimbs for other things, we start to see this cantilever. So if you have this longer tail, that's going to help balance the forces here and also allow attachment sites. So look at all these attachment sites that help hold this neck up, this arch here, and then also we would have muscles to help hold this up. This also, when we see this firmness of the vertebral column, again, this was helped to be maintained by a network of strong ligaments and muscles that are attaching. So this is really cool too. When we just look at the bones, we can really think about what's going on and be thinking about what other structures like muscles are being attached to it when we're going to get into the muscular system next week. So when we have this neural spine, again, this spinous process sticking up, the angle of this receives the forces, right, at the least stressful direction. So what in the world am I talking about? So basically, if we can think about having these muscles on this, so the muscles are going to be pulling forward, the force is coming this way, and the force is going to come this way. And then it's going to result in the resultant force down into the bone, okay? So this angle, again, this is going to be parallel with the resultant force, okay? Now, if we extend the height of the spine, the taller the spine is, the height is going to be proportional to the mechanical advantage or leverage, right? So just like a prior like if you're going to pry bar or something like that, the longer, the more force you have. And again, this is also for more muscle attachment. So we can now look at it with a bison, okay? So here, again, we have these tall neural spines. You can think about the muscles being attached, helping to hold up the head here. And you can see these arrows with the forces, okay? And I'm going to go ahead and let you pause this here so that you can read this explanation a little bit better. So again, when we're thinking about these neural spines, again, the height of the neural spine helps to tell us the relative weight that has to be supported by the vertebral column. Here, this is a skeleton of a pelycosaur, okay? With most of the neural spines of similar height and orientation versus the bison. And then not all pelycosaurs had these short spines. We also had these really tall. This is a Dimetrodon with these really tall spines, so probably for muscle attachment to help hold up the head. All right, we're going to end here, and the next video we'll go through the appendicular skeleton and fins and limbs. See you soon.